ahead.
Yeah. Okay, so thanks for the introduction. Yidi. So today I'm going to talk about the statistical methods for analyzing gene expression data in the single cell era. So let's begin with first do a little bit introduction about me. So I'm a new postdoc of ethics of the Australian green industry. Which is led by Amy, and we all have like Fonty and Patrick in the lab, and I'm Yidi. Yeah. So, before I start this job, I did the PhD in applied statistics and biometrics, from the University of Melbourne, and I was supervised by Professor Kim and Nicole. So the course lab does a lot of work in developing like statistical methods for like data integration. That is how you got data of different modalities and how you like jointly analyze this data together. So it's multi-omics data, for example. But my research focus is more on a single cell. I will talk about that later. And I mentioned that's going to be the main topic of today. And before my PhD, I also did some like analyzed in transcriptomic analysis, where it mainly focused on analyzing the structure of the transcripts, like finding the alternative splicing and isoform transcripts. Yes. So today, we're going to just talk about the methods that we have developed for analyzing the single cell expression data. So we have Sincast that is used to impute single cell data, and we have StableMate that is used to find generalizable association between gene expressions so that we can infer like regulatory events happens in cell. And finally, we have NeighbourNet, which is the method that we can use to infer cell-specific gene regulatory network. You may not know what does that mean, but talk about that later. So let's first go through some of the biological background. Of course, our body have like diverse cell types. We have like muscles that can power the movement. We have neurons that can transmit signals, and we have immune cells that can defend against our body. So each cell type or even different cells have very distinct functions. But despite their functional diversity, almost all the cell types or the cells contains almost the like similar or even the same genetic information that is encoded by DNA, so which is known as the dictionary of life. So the major question that we want to ask is what actually contributes to this functional diversity, right? And the one big part of the answer is gene expression. And gene expression basically is the process where the genetic information encoded in DNA are converted into functional products such as mRNA and proteins through the process of transcription and translation. So different cell types or even different cells express genes very differently. So actually, it is very natural that for us to like profile the gene expression of the individual cells to study their cell function, because we know that gene expression contributes to the cell function. And luckily, now we have modern technologies available to measure gene expression level in a very like high speed and cost efficient way. That is, for each sample, we can measure like thousands or tens of thousands of genes simultaneously at very low cost. So basically what we do is that for our individuals, we just collect samples from their individual tissues and then send these samples to the sequencing machine to measure the mRNA abundance within each sample. So normally when we talk about this mRNA abundance, we're just counting the number of transcripts within each sample. And finally, what we'll get is the gene expression matrix, which is a count matrix, where we have each sample being measured on tens of thousands of genes. Okay, that's basically the gene expression. So what I can do with gene expression data, and here I just introduced the two most commonly done analyses, which is the co-expression analysis and the differential expression analysis. So for co-expression analysis, we just measure the statistical association between two genes expression. And by doing this, we can do like gene clustering by their expression so that we can identify the gene modules that could represent some functional module that regulates gene and cell function. And we can also do this gene regulation inference, that is some kind of like the causal inference, like from observed expression data, we infer which gene is regulate which. So this is gene regulation inference. Or we can do differential expression analysis. Basically, we just compare the samples of different conditions, and we just kind of like correlate the changes of the expression profile to their functional changes or like observed phenotypic changes. But what we just talked about is what we call about a bulk sequencing. That is, we just measuring the expression within the sample. So this sample could be considered as this sample expression could just be considered as the bulk aggregate expression of many single cells, right? So it is not actually at the single cell level. But today the technology has advanced to a level about like 10 years ago, even earlier, to a level where we can now measure single cell expression. That is, for each sample, we can disassociate the sample tissues into individual cells, and we measure gene expression level in these individual cells. So we got the gene expression data, or we substitute samples with cells. So we often get like thousands, tens of thousands, and now today we have millions of cells in a single data set. So it's very huge data set. So what do we do with single cell analysis? So we have the gene expression matrix. What we do first is that first we just do a dimension reduction. And here is the dimension reduction technique called UMAP, which is nonlinear dimension reduction. And based on the reduction, we do some clustering and annotation clustering of cell to generate cell clusters and annotate these cell clusters by their their expression profile. And the annotation that we normally do is like cell type or cell state. For example, in this data, we have progenitor cells, and we have, so this data set is the data set about blood cell development or hematopoiesis. Or we can do this trajectory inference, that is, we find the development trajectory along this gene expression space. And of course, the common analysis that we do with the bulk data, the sample level data, can also be applied on single cell data, the differential expression analysis, the co-expression analysis, and etc. Okay, but as you can imagine, the single cell gene expression would be much noisier and sparse compared to the bulk expression data, because bulk expression data is the average expression. So it is inherently more stable, but it doesn't necessarily mean it's very like high resolution. But a single cell data, within each cell, you have much less genetic material. So this expression is more likely to be influenced by a lot of like technical factors. For example, the contamination, that when you do the sequencing, there could be get contamination from the other cells. Or like in sample preparation, you could like, if you don't prepare a sample correctly, you could stimulate the cells, which make the cell into a state that you don't want. And when we talk about sparsity, it means that single cell expression has a lot of zero because there's much less genetic material that is being measured within individual cell. Okay, so that's the background about what is single cell expression. I hope that's clear. Yeah.
Your right-hand side plot, is it for just one sample and then for many cells, or that for samples?
For this one is just one sample, but of course you can analyze multiple samples together.
Okay.
Yeah. So each point is a cell from a sample.
Okay.
Yeah. So first, I want to talk about the first project, which is Sincast. Sincast for single cell imputation annotation. because you can imagine if we have a very noisy and sparse data set, the downstream statistical analysis could be very difficult, right? So the first attempt that we want to that we have on single cell data is single cell imputation to make the robust downstream analysis. So to illustrate what's the statistical hurdle behind the single cell data analysis, I want to use this example of cell type annotation. So when we consider a cell type annotation problem, it could be just considered as the classification problem where you have the reference data with known cell type labels. And then we can just use this reference data to predict the cell type labels or cell state labels of the query data. So if you have any experience in doing single cell data analysis, you must have heard about this single R package, which uses bulk transcriptomic references datasets to annotate single cell data. So you can see this is the download stat of the single R in 2025. See this, it is very, very popular method even till today, but it's developed like 6 to 7 years ago, I think. But the problem is whether it's a correct approach to annotate single cell, because you know there's a very strong technical difference between the bulk and the single cell data.
And just to clarify, the reference ones are validated experimentally?
Sometimes it's validated experimentally, sometimes it's just like manually annotated single cell data.
Okay.
So here cell type annotation, not just you can do cell type annotation by classification problem, but also sometimes people just like find the differentially expressed gene of each cluster and you manually like search for literatures and annotate that cluster. But that's actually the second procedure. The manual annotation is more used more in today's research.
Okay, I guess I was wondering how correct the reference is.
If it's
a different philosophy. The old days, you had marker genes.
Yeah.
If the marker gene was on, then you say that's the cell type.
Mm.
Now we have a whole
yeah.
transcriptional profile, and you could call it a big cluster and then
Yep.
bottom up, create a cell type that doesn't have a name,
Yeah,
but
okay.
it's surrounded by a lot of genes, and then people say, well, what's the name? Marker gene.
Mm.
There's really different approaches.
Yeah.
I don't trust the reference.
Yeah. Yeah. So, but in for example, I come from a background in blood cell research. So people just do flow cell cytometry. Yeah, and they just, yes, outside of the cell surface. And actually these markers are often for example, the immune cells that is used to recognize pathogens or communicate with each other. So they use these kind of like cell surface proteins to classify cell types. But still, even in today's wet lab research, they still use this approach, do flow cell cytometry, and you confirm whether the classical markers is present in that cell or not. Otherwise, you can't just claim that's the cell type you want. Yeah. Okay, so let's just put aside the single R problem. At that time, the cell type annotation is very hot topic in single cell, and everybody just want to do this kind of research to build a better classifier. But actually, we also want to do some research on that, but we attack this problem in a very different angle. So instead of developing a statistical method for classification, we want to build a integrated reference that contains rich biological information to make comprehensive annotation of the single cell. So this was the work that was led by Dr. Paul Angel and Professor Christine Wells from the Center for Stem Cell System in University of Melbourne. So what we did in this project is that we developed a very simple and efficient computational framework to integrate samples from very diverse backgrounds collected from different sources. And as one of the results, so this is an interactive PCA. As the one of the result, we build a myeloid cell reference, and we call this as a myeloid atlas. And it contains 900 bulk samples collected from like 44 different studies and containing 22 different cell types. And these cell types are cultured from like 3 different cultural environments and derived from 3 different progenitor types. So it's a very like complex and heterogeneous data set. So as a statistician, you can appreciate how good this kind of reference is. Right. Anyway, so now we have a good references. We build some good references, and now we want to use these references to annotate some single cell data that we're interested in, and we want to make some biological discovery based on our newly like constructed references. So what we did here is that we collected one blood single cell data. This is a AML, which is white blood cancer. AML data set of single cell data set, and project this single cell data onto the PCA of the blood cell reference. So you can see in this projection, the projected single cell marked as courses are clustered at the middle of the atlas. However, the cell type direction, for example, here is the lymphoid cells, and here is the reference lymphoid cells, here is the myeloid cells of the reference, and here is the myeloid cell of the query. You see, their cell type align in the same direction, but still they're underfit the reference. So this observation suggests that the 2 data set has biological variation in common, but they share very little variation in common in terms of the like technical differences. So you can imagine if you use like annotation methods such as single R to do this kind of annotation, how wrong it could be. So we think that the major problem underlying this disalignment between the single cell and bulk is because the single cell data contains too much zero. So the first idea that we have in our mind was to impute the single cell data and then do the annotation. So fill up the zeros. And specifically, we want to impute it in a way such that we can keep the continuity of the single cell identity on the manifold of the expression space. Because single cell, the reason that we study single cell, the big part of the reason we study single cell is that we care about this kind of like continuity of cell, like for example, development of cell state transition. So
people do it is they just don't try to use all 20,000 genes. They just say here's the 3,000 most abundant genes that don't have a lot of this.
Yes, actually, that's very good comment. So actually, this reference data set only contains 3,000 genes. Yeah. So we also, when we build this reference, we also did like filtering and the most important genes. Okay. So the imputation approach that I want to take is called light network propagation. And so the underlying this network propagation algorithm, the core is that we need to build a affinity graph of cells. It's a cell-to-cell affinity graph that measures the similarity between cell expression. So each node here represents a cell. And the way how we make the imputation is that we net the cell to communicate and exchange their expression with their neighbors on this cell-to-cell graph. And this imputation is done in a step-by-step manner where in each step, the single cell expression is updated after its communication with the other cells. So it can be done in multiple steps. And here is the first imputation method that we tried, which is called magic imputation. And it was a very popular imputation method at the moment. And this method was based on network propagation. So here I'll just give you an outline of the magic algorithm. So most of network propagation method will be very similar to this, but they mainly differs in how they construct this affinity matrix, because this affinity matrix actually describes the describe the distribution of the cells on their expression manifold. So actually, it is very intuitive. So in the first step, we just perform PCA on the gene expression data and calculate the distance between cells on this PCA space. And then we apply a adapted Gaussian kernel on the distance matrix to construct this affinity matrix, which is essentially a KNN graph in which each cell is connected with its k nearest neighbor, k nearest neighbor with the most similar expression. So this measures similarity in single cell expression profile. And then we just symmetrize the affinity matrix and row normalize the matrix such that the row sum equals to 1. So this is a Markov transition matrix. And then the propagation is simply done by applying the exponent of the Markov transition matrix on the gene expression matrix. So basically, we can think about we're taking diffusion random walk of cell expression on this affinity graph. Okay.
So in some sense, the neighbor is determined by its actual expression type.
Yes.
It's no external information. Okay.
Yeah.
Okay.
Yeah, but you know, nowadays we have like spatial transcriptomics,
Yeah.
where
so
we
that-
actually have, yeah.
What the neighbor is spatially.
Yeah.
But yeah, so this is implicitly assuming that if they are close in expression, they must be closely related to each other.
Yeah, but actually, if you just look at the spatial expression of the single cell, you see that very close expression could like very far away from each other on the spatial.
Yeah,
issue.
yeah, that's what I was thinking, because this is making that assumption here.
Well, you need to understand if you have your skin,
Yeah,
there's goes to the regular cell and then up to hair cell, or the sweat gland cell,
Yeah.
and now back to the cell.
yeah,
Yeah.
There's clusters.
yeah, true.
What's the distance metric here, Euclidean distance? Is there something?
Yes, Euclidean distance. So
Yeah,
it's
okay.
very simple, like Gaussian kernel
Yeah,
applied to the distance measure. But the smooth the kernel width is adjusted for each neighborhood.
Yeah.
So it's called adapted Gaussian kernel.
what?
Can I ask? Can you hear me?
Why the distance is calculated on the PCA rather than, I guess, the directly, I guess it's a single cell is just ones or zeros when it comes to actual data.
Now you often do like some kind of transformation before you do the PCA. So you have const data, you normally, the most common transformation that you do is log transformation.
Yeah.
Yeah.
And then you take PCA on that, and then
Yeah.
you take the distance.
Yeah. So basically, this kernel function that I constructed is actually the diffusion kernel. So there is some kind of like a geometric assumption behind this. So even though it's Euclidean distance in this PCA,
Yeah.
but here the affinity matrix, you can consider it is a kind of measure of like distance on the manifold.
Yeah, okay.
Hello, can you hear me?
Yes.
I just wanted to double check. Maybe you mentioned. So what's the input like? How do you pick your cells? Is it like significantly expressed genes, or for this when you start PCA? Basically, what goes there? What kind of matrix?
So the input is just gene expression matrix.
Cells.
Yeah, just any
Yeah.
expression. So you don't filter for highly expressed. So what I mean, even if the gene expression is, I don't know, 0.1, you still consider it as an input for this.
Yes. So because it's imputation method, its function is to like get zero expression to some kind of like imputed value. So that's the goal of that method, right? So it doesn't care about whether it's no expression or not. Yeah.
Okay. Yeah. And so basically, how big is your what's the dimension of your matrix? Like, I don't know, 1,000 by something? Yeah.
So this matrix, I think this method works very well for like 20,000, 30,000 something cells. But of course, if you're like, because you are constructing a distance matrix, right? This distance matrix can explode and taking up too much of memory usage when you're constructing it. So
Okay.
it can't be too large because that was developed in 2018. So we don't have that many single cells in one single data set.
Okay, thanks.
So in the plot,
Yep.
Those are like two genes,
Yeah, two genes.
and then as you go more interaction, you
Yeah,
can
yeah,
capture the
yeah,
plot expression.
yeah. Yeah, so because you take more smooth, you make more communications and it more closely. Yeah.
Yeah, what's different between each time step? Wow, it just sounds like one step.
No,
Yeah.
so here, T is this time. It's called diffusion time. So just imagine each T, from 1 to 2 is like moving, make one step of movement and then make a second step of movement. Yes.
The raw data is gonna have maybe 10,000 cells by 10,000 genes. And you calculate the distance, and then for a lot of those 10,000 is zeros.
Yeah.
And then after the first round, you put in a little something for zeros.
Yes.
And then you calculate again, and you calculate again until you fill in those missing values to the sum.
Yeah, that's very correct.
I was wondering that you put MT into the power TX there. Then is it possible that you transform it once, like using MT, and then repeat the entire process again to get the second result?
So basically, if you choose MT, it's basically you multiplying M for T times. So
Yeah.
that's what you mean, right?
Instead of doing that,
Yeah.
you get your X hat and then repeat the entire process again.
Yeah, so he's saying recalculate M. A
Um,
bit time consuming, maybe. I don't know.
Yeah.
You can do this, but actually here your PCA is actually like in the graph informed. So it's kind of weird if you do that. Because you already kind of like applying the like diffusion kernel on the expression matrix. So your matrix has already been projected onto that space. And now you do the PCA. So it's already been informed by that transformed expression matrix. So I can tell you like if you do this, your variance of cell after imputation will be very low because you are over smoothing the data.
Okay.
You don't drop any.
Now you drop pieces. They take the most important pieces.
All of it, but
And what's the stopping criteria of that? What T would you choose?
They don't give a correct answer and a exact answer. Normally like 3 to 4 should be enough. Otherwise, it's gonna be too terrible. You see the variance has reduced a lot during this kind of iteration. So okay, now let's do the magic imputation with like suggested input of the magic algorithm, which is 3 steps of imputation. You see, it is much better. The projection is much better.
Well, what's the, like, how do you assess beta? Like in
Because we know these cells should have the similar cell type in the similar cell state.
okay, so it's kind of informed by the clustering looks right, given
Yeah.
informed from what you
Yeah,
see,
yeah.
The better
So
coverage?
and but
Yeah.
okay. It's a separation that you can see.
Yeah. Yeah, so it is good, but it is not good enough. Okay. So because of that, we're trying to develop our own imputation method, which is Sincast. So basically Sincast is based on like the network propagation, so the framework shouldn't be different too much. But we identified a very big problem with the magic imputation. So just consider now we have the this k-nearest neighbor graph or the affinity graph, where each cell is connected to its 5 nearest neighbor. And now let's just look at this cell 1. So if you look at this kind of like distance to affinity function, we will find that this cell 1 will have a very high affinity on itself. But its affinity with the other cells even no matter whether it's the same or different cell types, will be very little and indistinguishable, right? And this is just because of the fast decaying rate of the Gaussian kernel function. And this is also one very common problem that we know in like curves of dimensionality, where your dimension gets too large, your distance difference is too small, so we can't distinguish between points. That's curves of dimensionality. So that happens in here, even you use PCA. So different cell types contribution on imputation of cell one would be similar to these two cells. So Sincast approach is actually very simple. For each cell, we just subtract its distance with the cell's distance with second nearest neighbor. By doing so, when we apply the Gaussian kernel function, we're kind of like focusing on cell one's local connection, but we still keep the distal cells like influence minimized. So it ensures that cell one at least closely connect to one of the cell, right? So that's one way that people solve the curse of dimensionality. Other than that, we also proposed the Bayesian approach to shrink the imputed expression data back to the original expression. So basically, we're taking the Bayesian average between the impute data and the original data because we want to avoid over-imputation. And in this case, we just said the original expression is actually generated from the imputed expression plus noise. So that's how we structure our Bayesian model. Yeah, so it's kind of like an empirical Bayesian approach, but I'm not going to go into details about this. So you see, Sincast imputation, magic, and before, apparently the Sincast imputation align with the reference best and has a lot of like variation even within the local neighborhood of cells. But you may say, as you just asked, how do you assess? Why can you say this is better, right? So here is the benchmark study that we did. So the data set that we use is this data set where we captured like 10 very distinct subtypes. So what we did is that we just aggregate cells of the same cell type into pseudo-bulk samples. And then we run PCA to build a pseudo-bulk reference PCA. We'll project a single cell onto the pseudo-bulk reference. So we kind of get the ground truth about what it should be. And here, the fainted dots here are the samples, pseudo-bulk samples. You see, Sincast align very well, will keep the continuity of cells between these clusters. But for magic, it's not very good. K-smooth is another like imputation method based on network propagation, and Saver is the deep learning method. Yep. What time is it? I think I need to skip the other. Yeah. Why the second? Because we only want one cells, because the third one could be very wrong. So the basic assumption on the line is it is that there is at least one cell that is very similar to your expression profile. But we don't know about the third one.
It's good for groups of cells.
Yeah.
Very rare cell types, you'll be the wrong thing.
Yep.
But as long as it's got little groups of cells.
Actually, we did one simulation study where still the pseudo-bulk projection, and we trace the projected cell center on this reference. So Sincast, if you just increase the propagation step, well, so this is the projection here is the DC6 cell. So this is the DC6 reference. You see the Sincast, it will stay close in this DC6, which is the real cell type here. But for magic imputation, if you improve this, like increase the propagation step, you will just move away towards this large population. So that's why we want to emphasize on this local connection. Maybe I don't talk about the biology here. Okay, so the next thing that we did is called StableMate, which is very different from Sincast. Where Sincast is used for cell annotation. Oh, I forgot to mention, within Sincast, we also developed a annotation approach. So it's a pipeline, not just a annotation method. So StableMate, it deals with another perspective of analyzing single cell data. It measures co-expression between gene expression. And specifically, we make this called generalizable measure of co-expression, and you'll find out what does it mean. And we want to use this kind of co-expression to infer like kind of approaches gene regulation events. Okay, so the actual kind of statistical association that we want to infer between gene expression is called stable association. So stable association by definition is kind of the association that is invariant and robust when the data set is subject to external perturbation that could influence the data distribution. So robust to perturbation is good because it can ensure reproducibility. And that's one thing that is missing in today's biology research, especially in co-expression analysis. And also, stable association have causal implication. Why is that? Because physical causality is a physical law that is mostly stable. So we use stable association to approach causality, but we can't say like stable association is causality. And this improve our interpretability of the measured co-expression. Otherwise, if you just use correlation, what does it mean? Okay, so let's just jump into the StableMate framework. It assumes the multi-environment regression framework, where you assume that you have data that are collected from different environments, but are measured on the same set of predictors, for example, genes. And the goal of StableMate is to find a set of stable predictor such that the regression model fitted on the response of interest are consistent across different environment. So just imagine that you fit model in different environment and you want this model to be consistent. So that's a stable predictor.
What is the environment in the context of cancer?
So for example, in cancer research, we have like pan-cancer research. Like we want to find the molecular mechanisms that causing like, not just, for example, non-cancer, it also causes like different cancer, pancreas cancer, like brain cancer, etc. So it can be cancer type, or it can even be cancer subtype.
Okay, okay,
Yeah.
okay.
Could it even be cell type?
Of course.
So you switched, I guess, maybe not everybody noticed, but I try to emphasize, now you've got genes across the top and samples down the side. So these single cell data sets have thousands, tens of thousands of cells and tens of thousands of genes.
Yeah.
So if you're trying in the first part cluster cell types, don't care much about the genes, you want to cluster the cell types. Now you care a lot about the genes and you don't care about the cell types, are just environments. You're trying to get the gene code expression. You just turn the matrix and analyze the other side.
Yeah, that's a very good point. But actually, other than the stable association, if you look at the counterpart, like the unstable association, it's also very interesting because that's the association, the genetic relationship that changes across cell type or condition. So it's also biologically meaningful. Yeah, but I didn't talk about that because it's kind of like biological. So basically what we want to do here is that we want to infer generalizable functional dependency between the predictors and the response. So that's what we want to do. And here I just show you the difference between a stable predictor of the response and unstable predictor of the response. And the stable ones, you see they have very like similar linear relationships, exactly the same linear relationships, but for the unstable one, the linear relationships differs by intercepts. But StableMate, of course, can be applied to different, like, can be applied with different regression model, not just linear model.
They're kind of stable where you just have
no random effect. Yeah, but maybe this can be an extension, maybe my next project. Yeah, so before I go into the algorithm, I just first want to show you what's the StableMate output should looks like. So here's the variable selection plot. And the input of StableMate should be a data that is being partitioned by some kind of user-defined environment. For example, here we use the breast cancer gene expression data, and we choose the environment to be the disease condition, the different breast cancer subtype. And the response that I want to predict here is the ESR1 gene expression, which is also gene response 1. So it's a very critical gene in breast cancer. So here on the x-axis, we show the predictivity of the predictor. That is how important it is in predicting the Y. And on the y-axis, we have the stability. That is how stable it is predicting the Y. So these genes over here, at the upper right corner of this plot, are the genes that are stable and predictive. So this is the genes that we want. And according to our stability assumption, these genes should be very closely related to the regulation of ESR1. So for example, if we check this ARMT1 and say this is 117 here and check it on the genome browser, we will find that these two genes are located closest to the to ESR1 at its upper genomic region. Here. So this is ESR1, which suggests that these three genes could be subject to the same kind of genetic regulation. So that's what we expect from a stable predictor. So it should be very closely related in terms of biology. I don't know how it is related, but it should be related. Okay, now go into the statistics. So the actual thing, the quantity that we want to infer behind the sense is the stable Bankit, which is the concept that is derived from Markov Bankit in structural causal modeling or Bayesian network. So for example, let's look at the left-hand side of the plot. So this is a causal structure that illustrating the data generation process of different variable, and just assume this response is colored as orange. So the Markov blanket of the response is actually the set of predictors conditioning on which renders other predictors independent with the response. Or more intuitively, we would just say that Markov blanket is the smallest set predictor. Smallest set of predictor that is most efficient. However, what if now our system is being perturbed by some environment factor? Now, if we just condition on this Markov blanket, for example, we will see that when this environment factor perturb the children of the response, conditioning on this response will establish the statistical dependency between these two variables, the response and the environment factor. So this means that your prediction will be confounded by environment. But this is not the problem with the causal parents of the response, because when conditioning on the parents, it will just block the information flow from the causal parents to the response. So that involves a little bit of causal reasoning. So just imagine that here you have two parents because they're all influencing these children. If you know what is the genetic information of the children, then you will kind of associate the genetic information between the parents together. So that's the intuitive here. So as you can imagine, if you have a lot of perturbation on the system, the stable Bankit would be just, so the stable Bankit is the most predictive stable set within the Markov Bankit, such that its prediction on the response will not be confounded by environment. So you can imagine if you have a lot of environment perturbation, the stable Bankit would be just equal to the causal parents. But if there's no intervention, the stable Bankit will be just equal to the Markov Bankit. So so our goal here is to find a stable blanket. And the way that we find this stable blanket is actually very like classical. We just do a like regression model and we find the set of predictors that like optimize the some kind of like objective function of the regression model. And this is a greedy search algorithm, and it is a stochastic selector. That is, if you run this regression model for multiple times, the predictors that you selected could be very different because it's random selection. But I'm not going to into details about what it what is.
So it's like leaving some predictors out, checking based on the objective function, what the actual response is, what the value of the objective function is.
Yeah.
But here you're just choosing a randomly.
Nah, so basically it's very similar to stepwise regression. So stepwise regression, you just choose the one predictor to add or remove from the model. This algorithm add or remove a set of predictor from the model, but it is removed in a random fashion.
Okay.
But how you remove it is a problem. So we did some work on like how to remove or add predictors in.
Okay.
So we do this stable blanket selection in a two-step manner. So in the first step, we just run this selector for multiple times to select an ensemble of very predictive sets of the response. So we don't care about the environment effect. But in the next step, we select stable sets within the predictive sets, within each of the predictive sets. And the reason that we can do this is because stable blanket must be the subset of markov blanket, right? So the objective, very easy. For the first step, we just want to minimize the BSC because it can be shown that the set that minimize BSC equals to the markov blanket under some certain assumption. And then in the second step, we just minimize the coarse validation performance, which is mean square error coarse environment. So we use the environments as partition. So we train the model on some environments and leave one environment out, and we just predict on that environment, and we want to minimize this as extrapolation error. So very intuitive, very simple, right?
May I ask, the first step you minimize BIC, is it you put all the regressor into the regressions, and then you minimize BIC by removing some of the predictors from the regression?
So minimize BIC, I mean that for each round of like selection, when I apply this selector on the prediction set, I try to minimize BIC. So each set is selected independently.
Okay.
But because the reason that we use this like stochastic selector is that it can easily because stepwise selection can easily converge to local optima. But for this stochastic selector, it will like approach more options. It's more like a random forest, like you have a lot of like different models constructed. So your model is more robust, right? And that's also the reason that we want to construct this variable ensemble. And because this, so BIC is very hard to optimize. It's not something that you can just go down and get it.
How many environments?
So so it doesn't, you can have as many environments as you want, actually. But in the previous the selection port that I show you were just 2 environments. But I do have like a case study where I have 8 environments. So how you code these environments could also be different. So there's a lot of things that you can play around, and I allow a lot of flexibility in this package. And the good thing about mean square error is that if there's no environment effect, the mean square error will guarantee that this stable set will be equal to the predictive set, but because you will just include as much predictors as possible. So that's why I choose this mean square error. And then we just calculate selection frequency in each of the ensemble, and we get this prediction score. Easy. And finally, we just want to make classification on which is like important selection, what is like interesting selection, given this predictive score. So what we do is that we add a pseudo predictor into the regression model, which is basically an index. It's not an actual predictor. It's an index where you can select during this process. But this can be selected, but it won't influence the model fitting, and it won't influence the objective score. So if the true predictor is selected more often than the pseudo predictor, it means that this true predictor could contribute to improve the objective function. So that's how we get this threshold value.
But this one threshold, or your pseudo predictor is there, no? So it's
Yep.
not.
This is pseudo predictor. And we also, of course, we have a interval.
Oh, okay. So it's actually okay. I get it. And then, so I didn't quite get how are you constructing it. So somehow it's not going to alter the objective function. Right?
Yep.
So it's not just like random noise that you're adding there.
No.
But you have some clever way of determining what this pseudo predictor is, such that you know it's not going to influence your response.
Just imagine that you have like 100 predictors, true predictors.
Yeah.
And now I have a 101 predictor, which is basically an index. There's no data associated with this 101.
But when you say index, what does it mean? So it just like
So it's just an index. So it is something, it's called X101.
yeah.
So that's it.
But if it's a predictor, it has to have values.
No. So when we select predictors, it's like random selection, right? So we could, a set of predictor could contain that X101. But when we fit the regression model, we don't use that X101 because we don't have it.
Okay.
Yeah. You understand what I mean?
Maybe not. Because if it's something, it doesn't have to have an actual value like in the predictor.
No, you don't have to. So basically, there's a set of predictors. For example, I have X1, X2, X101, right?
Yeah.
So we fit the regression model with this F X1, X2. But we call this regression model is fitted by X1, X2, X101. So with this, we have data. So we fit this regression model, get the objective function from, for example, BIC score. But we pretend that this regression model is actually this regression model that includes all these predictors.
But what? But there's no okay. So there's
Then you assess the performance of that X101 in the form, right?
We assess how often this X101 is contained in this independent selections. So we still make selections.
I think I understand. So you just randomly put like your pseudo predictor in there.
Yeah.
Yeah. Okay. I understand. Yeah. Okay. I see.
Mark.
Yeah. Okay. I think I follow the idea.
Yeah.
And so that's and then use that for stress solving.
Yes.
Okay.
Yeah,
That's interesting.
so basically we're comparing the distribution of the pseudo predictor with the distribution of class predictor. So each predictor should have a interval. But I don't illustrate it here, but it's just a mean of the selection frequency.
Why is this pseudo predictor so pretty high precision, pretty stable?
We don't say because the predictivity and the stability is assessed according to pseudo predictor.
Yeah.
We don't say it's predictive because its score is very high. Sometimes it's because of data. So for different data, the interpretation of the score could be very different.
Yeah. I mean, so this is the part where the scores don't have actual interpreted meaning.
Yeah.
It's only was relative to the pseudo predictor.
Yeah.
Yeah. So you shouldn't think it's high just because it's up there. Everything is determined with respect to that.
Yep. Yeah, so basically that's StableMate. I will skip this benchmark steps. But I got
sorry. So maybe in that sense, because you just finished the StableMate, and I know you have your last one as well. Maybe. Yeah, I think so. It's certainly a lot, and I think maybe something to emphasize is that all your methods that you've introduced is actually on your GitHub.
Yeah.
So for those people who want to use the code, it is available online as well. So maybe we won't go through into the NeighbourNet. And maybe
Well, I just give you the idea about what is it. So NeighbourNet is you infer a network for each of a cell. Actually, I want to talk about this biology behind it because maybe like Patrick was interested in this next time. Yeah. So yes, so basically you have for each of the cell you get this kind of network where you can play around with, so you can do a lot of analysis with this network. So basically when you analyze single cell data, you analyze the expression, but now you can analyze the network of each individual cell. Yeah, so that's something that can be very biologically meaningful.
and we'll make sure you have time for another time, I reckon. Yeah.
Oh no, thanks.
Well, thanks very much, Yidi.
Yeah. I do want to say, could we have a group photo?
Is this the image that you have? Yeah, good seeing you. But the online people just went off. I guess they had to.
Good. Thanks for getting us together.
All right, thanks. See you next time.
Thank you.
You're up next time? Yeah.
Yeah.
I'm going to, that's great. Good job.
Thanks.
Right,
Nice
Thank
to
you.
see you.
Ash is coming now. Oh, yeah.
Well, I mean, you got asked questions on the way, but was there further questions?
Start to your environment, like
Yep.
Environment counts the time in this instance.
Yeah.
I think we need to disassociate environment from what we think in the plant world.
What is red? What do we need? You know, like the environment predicts it, and then nothing else predicts. Like when you introduce the non-conforming.
Environment is something that you don't observe. So it can be anything. For example, here we have a case study in metagenomic data. While here the environment is the different study cohorts. So here is just basically a meta-analysis, and you find the stable predictor that are predictive in different cohorts that around the world. Right? So we don't say like environment is predictive of something. We just use the environment as the approach for us to analyze stability to see whether there are similar or different across environments.
It's used very differently in the context of often when we call, environment in the plant, which is more like actual like environment plant. Whereas this is some kind of discrete variable that separates the samples into what we think is more similar to each other.
Yeah.
Yeah.
Yeah, grouping factors.
Yeah. I don't know why. This is the part where it mind boggles me when you call it environment, which is probably common in the field, right? Because again, coming into agriculture, it's actually not really like it will have a different meaning when we say environment.
Not actually people don't understand what is environment even in that field. That just it is the term that is invented by the original statistical paper, the theoretical one.
Yeah, because it'd be easier if they say some kind of grouping factor instead, right? And then that feels like more general enough to say it can be, because I wouldn't say cohorts are environments. But I guess cohorts here is that they're environmentally living in a similar place. And then the example that you showed was different cancer types. Again, that I would not think of that one as an environment. I guess that's
because in our paper, we also care a lot about the unstable predictors.
Yeah.
Because this one could be the one that differs across different like disease type. So that's more deserves like investigation compared to stable predictors. But stable predictors, like in the sense of like selecting like breeding markers, it should be more useful.
Yeah. Yeah.
So, in the step one, you said you randomly select a set of predictors from the entire set of predictors.
Yep.
And then, is it you then drop some predictors based on the minimized PIC of that set?
So it's same as like stepwise selection. You add or remove predictors to reduce the BIC value of the current model.
So how does the algorithm know which one to add and which one to drop? Because there are
So it's randomly selected. So for example, you have 100 predictors, but we just randomly sample some predictor set out of it, and to assess the performance of removing or adding this predictor set. So the major difficulty in this algorithm is how do you determine this kind of sampling approach? How much you should sample?
yeah.
How like efficient, how like diverse you should sample? Yeah. So that's why I don't go into details here.
So it's not okay. I see. It's not covered here.
So
It's
you've
not.
got some default in your software at the moment.
Yes.
Of what it should be.
Yeah.
Yeah. Because I wonder how much things changed. I don't know if we ended up assessing that at all. I mean, it's opening up a can of worms, right? Because you can keep on going and fine tuning everything.
Yeah, that's right. So I didn't do a very comprehensive benchmark on that, but
Yeah.
in experience, it doesn't affect too much. You just like do like slight tuning. Yeah.
Yeah.
And this environment assumption gives you the benefits of not caring about it at the first step, right? You can just minimize your PIC and then second step you take care of the environment.
Yeah, that's a very interesting, actually very interesting phenomena. When you do regression model using different environments, you just concatenate the data of different environments together and you do the regression model.
Yeah.
It can be proved that the regression model will just converge to the causal parents. For example, if you just use the linear regression, your like unstable predictors will simply be shrink to zero if you have enough environments and enough like environment effects.
It kind of makes sense.
Yeah.
Statistically, it makes sense.
Yes. So it's basically you have more like data from different sources. Your prediction model should be more robust. That's the idea, but it can actually be proved like in statistical theory that this is the case.
Okay.
And there's not a lot assumption behind this proof. It's very interesting.
It's equal to zero.
It converge because if you think about you estimate the parameter like more accurate with more data set with more observations.
Yeah, but it's equal to zero if I guess they are unstable, right? Because if they are stable, don't they converge to some non-zero value instead?
It means that the beta of that parameter is zero. The true value is zero. So when you have more observation, it should converge to zero.
But that means that it has no influence on the response, right?
Yeah, because it's unstable.
Yeah, if it's unstable. Yeah. So if it's stable, it should be non-zero. Yeah.
Or it could be somewhere very close to zero, even though it's stable. That's small.
Yeah. Okay. Very good. There's actually quite a lot of things I want to ask, but I will close up because it's over time.